Okay, let's dive right in. Today, we're putting on our detective hats and stepping into the microscopic crime scene. We've got a mystery microbe on our hands, a real culprit causing some trouble, and it's our job to track it down clue by clue. This is a real-world case of CSI microbiology.
So here's the situation. We've got a patient sample, and inside, something is growing. But what is it? And more importantly, what's it capable of? To figure that out, we have to start an investigation. And you know, every great detective story starts with that very first piece of evidence. So, our first move is to get a good look at our suspect to build a basic profile. We're heading straight for the microscope to see what we're really up against. And there it is. This is our first visual, and it comes from a classic technique called a Gram stain. It's one of the first things you learn in any microbiology lab. And what do we see? We've got a whole sea of these tiny, pink, rod-shaped organisms. Okay, so let's break down the lab report. We are officially dealing with gram-negative bacilli. Now, the pink color tells us it's gram-negative, and bacilli is just the fancy science word for rods. It's a great start, for sure. But there are literally thousands of bacteria that fit this description. We need a much more distinctive clue to narrow this thing down. All right, let's move on to our next piece of evidence, because this is where things get really, really interesting. We took the bacteria and let it grow on a petri dish full of nutrients. Now check this out. On the left, that's what most bacteria look like when they grow. Nice, neat little colonies. But on the right, look at our suspect. It's not staying put at all. It's spreading across the entire plate in these weird wave-like patterns. This is not normal behavior. And for us, this is a massive lead in the case. This incredible behavior actually has a name. It's called swarming motility. It's this super coordinated, rapid movement across a surface. And here's the key. Only a pretty select group of bacteria can pull this off. So our huge list of suspects, it just got a whole lot shorter.
And this is the big takeaway right here. When a microbiologist sees that swarming pattern, their brain immediately jumps to one group of bacteria, the genus Proteus. Just like that, we've gone from thousands of possibilities down to maybe a handful. But we still don't have our guy. We need the exact species. So, to get that final ID, we've got to go deeper. It's time to put our suspect through a battery of biochemical tests. You can think of this as a chemical interrogation, designed to make our microbe reveal its unique metabolic fingerprint. Our first major test is the triple sugar iron test, or TSI for short. This cube is packed with 3 different sugars and an iron source. Now, depending on what the bacteria eats and what gases it spits out, it's going to change colors in a very specific way. This chart here, that's our decoder key. And here's our result. The top part, the slant, is red. That's alkaline. The bottom, or the butt, is yellow, which is acidic. So in lab speak, we write that as K/A. But look at that dramatic black color all through the tube. That is a dead giveaway for the production of hydrogen sulfide gas, H2S, a very, very specific profile. But wait, here's the plot twist. While this result is super specific, it's the exact same result we'd get for another one of our prime suspects, a close relative called Proteus mirabilis. They're almost identical. Our interrogation isn't over. We need one more test to tell these two apart. Okay, this is it. The whole case comes down to one final test, the indole test. One of our suspects will be positive, the other will be negative. This is the moment of truth. So let's look at the files on our 2 main suspects. They both swarm. They both produce H2S. They're both positive for another test called urease. I mean, they're practically biochemical twins. Except for one thing, the indole test. Proteus mirabilis is negative. So, what will our culprit be? So this is it, the million-dollar question. After all these clues, all these tests, what is the one piece of evidence that is finally going to crack this case wide open? And the result is positive. Our suspect is indole positive. Boom, we've got our culprit. The mysterious swarming bacterium is Proteus vulgaris. Case closed. But, you know, identifying the culprit is only half the battle. Now we need to understand why this all matters for the patient. What's this bacterium's motive? What kind of trouble does it actually cause? Hey, remember that urease test where both suspects were positive? Well, this is it. Our Proteus vulgaris turns this entire tube a shocking bright pink. And that isn't just a pretty color. It's a sign of a really powerful enzyme at work. And that enzyme is the key to understanding the trouble it can cause.
So here's how it all goes down. Proteus vulgaris pumps out that urease enzyme. That enzyme finds urea, which is a waste product that's always in our urine, and splits it into ammonia. Now, ammonia is super alkaline, which makes the urine way less acidic. And in that high pH environment, minerals start to crash out of solution and form hard, jagged kidney stones called struvite stones. This bacterium literally builds rocks inside your body.
And that's not all it does. This microbe is notorious for causing nasty, complicated urinary tract infections, especially in people with catheters. And if you don't treat it, that infection can spread to the bloodstream and cause something called urosepsis, which is absolutely life-threatening. So getting this ID right, and getting it right quickly, is critical.
So here it is, the final rap sheet on our culprit, Proteus vulgaris. We followed the evidence all the way from its basic description as a gram-negative rod to its telltale swarming signature, and we used those final indole and urease tests to lock down its identity and understand just how dangerous it is. A complete forensic profile.
And that leaves us with one last thought to chew on. We've just seen how a single type of bacteria can act like a tiny, stone-building force inside a person. It really makes you wonder, doesn't it? What other incredible, complex, and sometimes dangerous microscopic dramas are playing out inside of us right this second?